So after we finish to run our core analysis, it will display it in a yellow bar at the bottom of the IPA main page, that it will show that it's finished, and it will give the name that finished. We can click here, or we can double click in the demo results. We're going to click here, and we'll activate the results created for us for this analysis. So as an overview, and for reproducibility as well, if we would like to overview what parameters we run, we can see it in here. In analysis filter summary, we see every parameter we set for this analysis. So in terms of, let's say, a descriptive sentences in a paper or publication that you use IPA, this is a good way to document and reproduce everything you run using IPA. So closing this, now we can go to the main part of the results. This section here is one of the most interesting ones, because here, this, as it says in the tab, it gives a summary. A summary in terms of the contents that are in the subsequent tabs at the top. So the first will be the top canonical pathways, in which we show the top 5 pathways in which the number of proteins is bigger. In other words, the top 5 pathways in where the list of proteins you gave to IPA is identified. It has been identified in that pathway. In this case, since we have so many proteins, so few of them, so many of them but so few in results, in terms of which of those proteins were considered significant, and we were very conservative in that, even though we try to be a little bit more flexible, and we are confirming by selecting those cutoffs that the 3 proteins we visualize using Lima are the ones that are significant in our filtering threshold using IPA. So even though that we don't have too many, we have the significant ones to work with, that is saying, pay attention to these 3, because maybe your scientific question, your hypothesis, was in those 3, and you need to work in terms of, and I repeat that term that some people in the community uses, as fishing, to try to connect your hypothesis with the results of the enrichment analysis you're doing using IPA in this example, and try to get an understanding on why that protein was significant in your hypothesis, what and why that protein was considered important for biological significance. So up to this point, we have been testing if they were significant statistically speaking, but they are biologically significant, and that's why we are using enrichment analysis to help us to answer this question in terms of, well, these pathways are the ones that I am studying in the laboratory. This function is very associated with something that the literature found as well, and we would like to hypothesize further this is connected with that. So IPA is mostly helping the researchers in that matter. So the first step is the top canonical pathways. It will show you only 5, but if we go to canonical pathways, if you have more than 5, in this case we have 6, let me just change this to horizontal, and in here, if we see the names of these pathways and we go again to summary, we see the same names. And these bar plots are clickable and interactive as well. So if we click one of them, it's saying, actually, if we click in here, it's saying that the ratio, for example, is 1/3. So it was expecting 3 proteins, and we gave it 1. Or that was, so we gave 1 because it's one what IPA identified from our dataset, but it was expecting 3. So, and the protein is significant in terms of expression, is significant in terms of statistical significance, and it's associated with NADH repair. And we can confirm that it has been identified in this pathway, and we also can check the pathway in IPA. If we want to open a pathway, we click Open Pathway, and it will redirect us to the pathway in where this protein is interacting with. And the nodes that are represented are other proteins that are interacting in the pathway, are molecules that are interacting in a step-by-step process of this pathway, and the ones that are colored red in our case, but is red because it's represented as a differentially more abundant or upregulated. If green, if red is upregulated, then green represents the opposite. It represents less abundant or downregulated protein or genes. And usually we can display the fold change below the name of the proteins. So to do that, we can edit some things here. So to edit this pathway, we can go to Overlay, Analysis List. It will just give you the overview of the number of proteins of our dataset that are identified for that pathway, and also it will give you the fold change, the p-value, all the descriptive parameters that we use to determine that this protein is significant. And we can say that this protein needs to show the fold change, p-value, and false discovery rate. So all this information is in here. Unfortunately, we cannot see this in here, but there is a way that we can edit this pathway even further as it is. We can say, well, I would like to edit this for a publication. I would like to edit it for something else. So this is the edition tool in IPA to edit molecules. And we can edit all sorts of detail of this pathway. So we can move things around, we can enhance the letter, we can change the font, the characters, we can change the legend, we can add legend in here, we can change the background. For example, personally, I like white background with intense colors representing our proteins of interest. In this case, we can show the legend in which it gives you the representation, what type of proteins are here, and it gives you as a legend. As well, it gives the opportunity to change the font. We can change the size. We can we can assign bold, italic. So it offers a range of edition tools that are good for publication quality material. Of course, something that adds more to interest in terms of, well, let's assume that this is already something that you're okay to publish. This is something that you want to publish as it is, and you can export this, and you can actually export the table and the images. You can export the table and it will offer you a very good resolution range from 72 to 600 dpi. Usually for publication quality, of course, the better the quality, the better for the publication. And when you're okay or satisfied with the parameters you selected, you say okay, you select the directory and type name of pathway in this scenario. So this one, name of pathway, and it will finish it. So we go to documents, to the directory where we did our statistical analysis, demo data, name of pathway. And it is saving, it's saved. So let's go now to our directory and would like to look to the pathway we saved already. So this is the one, name of pathway. And as you can see, a very good resolution quality that is ready to publish. Of course, you can edit it more. You can change as well the sizes of these nodes, of these icons, as big as you want, as smaller as you want. You can change as well the size of the values represented in each one, and so on, so on. So there's a plenty of options for you to edit pathways, and not limited to pathways, but networks as well. So the one that we previously discussed using the network interactions, we now can as well edit that network as well in here. So, and it doesn't limit it as well to the list of proteins we are providing here, but also it will provide the information of compounds that are demonstrated in the pathway. So that is only, and most of this information is only extracted from a single tab of the main results page. So let's just close this in here. Let's close this as well. All this information that was shown to you is from one single tab. So let's go to summary again, and now we see top upstream regulators as well. So this one shows you the top of regulators, 5. It will show you the top 5 regulators of this protein, and networks as well associated with proteins, microRNAs as well, compounds that are associated with that protein. Diseases, this is something very used, very implemented, very well studied. Most of the analysis, bioinformatics analysis associated with data sets that we work in collaboration with other investigators are related with diseases or pathways associated with a group of proteins under the study. And most of them appear in here that are also at the top. Molecular functions, physiological system, and developmental functions. Most of this terminology derive from gene ontology annotations. So they look more standard. They are standard terms to categorize genes, proteins, and compounds into a more defined terminology of functions. and actually usually ends up being a cascade of terms in biological process, molecular functions. So that's some sort of summary representation that I'm showing here. Top toxicity functions that are associated with. And of course, this is one of the most interesting ones because it shows you, if let's say that you have thousands of genes or thousands of proteins, and it will give you the top 20 upregulated or top 20, top 10, top 10 up and top 10 down, but in total 20 deregulated genes or differentially abundant proteins associated with your dataset. And something very interesting is now let's go to diseases and functions. This one shows you a heat map of the number of proteins in your dataset that are associated with functions. The darker the color, the more number of proteins or genes that are associated with that function. The lighter the color, the less proteins or genes are associated with that function or that disease. Most, as I described before, are functions from gene ontology annotations. If, for example, all of this is interactive. So let's say that you want to study apoptosis. Or let's say, as we were describing in the previous video, that these proteins are associated with some electron transport chain or redox reactions. We can search that in here. Let's just increase this a little bit. Then here you can see a list of all the functions associated with the proteins that you have. We can sort them out by saying, well, I would like to have only those that have all the proteins that are significant. Well, this is a way. You sort it by the number of molecules, and we sort it from higher to lower number of molecules, or number of genes, number of proteins, number of chemicals. How IPA interprets molecules is very ambiguous because it's not, well, the data that you have, they know already what they are. It's mostly based on, well, this is the information we have, and this is the information we are going to provide to you. But it's not that they know your hypothesis testing. They don't know that your scientific question is responded based on the list of proteins you have. You're answering that questions based on the annotations they are providing to you so you can get a better understanding of the association between the statistical significance and the information that provides IPA for you. So that's why most of the functions, let's say that you want something associated with some specific function, and probably you don't see it in here, is because IPA doesn't derive from your hypothesis testing. IPA derives from the amount of information that it has in store in their knowledge base based on the list of genes and proteins that you provide. So let's search for cell death, elimination, well, metabolic pathways. Let's search for glycolysis of cells. And we can see what protein is associated with that function. This tab in here is not pathways, these are functions. So if you want, you can display this as well as a network. Let's use glycolysis of cells, and let's say that I want to display this as a network. You can display this as a network. And here you see that GDAPTH is associated with the glycolysis of cells. You have up to 5 annotations The number here, it says the list of annotations that is associated with interaction. So in red, this protein is involved in the glycolysis of cells. Of course, we can search for those annotations in view relationships, and confirms the number of annotations it has, up to more than 5 literature papers published based on this information. So that's good because it confirms that this is literature based. And something as well that appeared is activity predictions. The activity predictions, or as IPA call it, molecule activity predictor. This, in a pathway, is very, very, very helpful. Let's go for a moment to the pathway that we were analyzing in the previous steps. Let's reopen it, and now we'll go to overlay, and now we go to molecule activity predictor. Of course, we leave it as default. So in here, these predictions are based on the literature that IPA has created its knowledge base. So if in the literature, the citations declares that protein A, in this example, not the example that I am giving in particular, just this verbal example. If protein A is upregulated in the literature, and in our information we have it as upregulated, and the literature is saying that when the protein is differentially more abundant, or if it's a gene upregulated, and in our data is upregulated or differentially more abundant, then leads to activation. That's good up to that point. But let's say that in your data is the opposite. In your data saying that that protein, the fold change that it's having is downregulation or differentially less abundant. So in that case, that protein or that gene is leading to an inhibition, even though that the literature is not saying that specifically, but it's inferred from your hypothesis. It's inferred from what you are reading and observing in the data, that you can infer that it's doing the opposite. And those predictions can be observed in here. So of course, most of the pathway is being activated due to the upregulation or more abundance of this protein present in this pathway, which is proportional to the activation of itself. And we have confirmed this as well because each of these nodes can be accessed as well and has annotations as well as the networks. So in this, human glyceraldehyde 3-phosphate catalyze the following reaction. So this literature is saying that when that protein is activated, it leads to that function. So let's assume for a moment that that protein is differentially less abundant or downregulated. Well, even though that in here it will still say in this annotation, the biological interpretation of what it's saying versus what you're observing in your analysis, it will be completely the opposite. So now you can infer, based on the information that you're seeing, that the result is completely different. Instead of activating, it's just not catalyzing that reaction and is inhibiting that reaction to occur. Therefore, the prediction here will be the opposite. So if we follow this legend, for now, if it's increased with more confidence, increased activation, or may lead to inhibition. Sometimes we see this when we see regulator analysis. We are trying to study what regulates what other things. And sometimes when something is upregulated or more abundant, may decrease the expression of something else and may increase the expression of other proteins in other genes. So this is the same example. So when something is more abundant or less abundant, it will have an effect. But based on the literature, that will have an impact on your results. But the results that you are getting may not always be the ones that are in the literature, but they are already reviewed by literature. So what you are getting is new data, it's new information, it's new results, it's something new, it's something that never has been done before. So even though that this information is in here as a canvas, the data that is, the way that the data is shown to us is new. No one else in the world will have this data the same way as you will have it. So with that in mind, let's close this. Let's go back here. So these kind of predictions are very helpful for pathways. In terms of networks, it helps, but it's not the same impact as it does for pathway interactions. So in here, as it shows, it doesn't give you any directionality at all. It's just pointing that GAPDH is interacting with glycolysis of cells, which by itself is a very fixed term because it's glycolysis of cells. So it means every cell it has to do glycolysis. That's the ontology term. But something that we can do is, as we done in the first video with EGFR, in here we can increase the number of proteins that probably are not in our dataset, that are in the IPA knowledge base, that may enrich these results. We can add more layers to this network as well. And of course, we can do that with the pathways, but it will be very convoluted and it will not have the directionality as it had before. So it will make sense to populate the networks instead of the pathways. Pathways are better to represent something more canonical, something more fixed, but when you want to represent something with not so much directionality, this is the way to go. You will want to go with networks. So we are going to do the same as we did in the previous video with this network as an example. And in here, we are going to be very less restricted. But since this is glycolysis of cells, probably we can try let's say cancer. And we always need to be sure that we are checking the nodes or the genes, proteins or molecules that we would like to analyze. Let's highlight this and then we click apply. Of course, there is a lot of them. So a way that we can reduce the amount of this, of course, we can undo this by Command and C. Is instead of giving all, get max of 10, just to get the max of 10. And then repeat the same process. And there we are. We're now having proteins, compounds, genes associated with this protein that are not in our dataset, but may enrich more, may give more information, something more that we can provide with the gene or protein we are under study. So here we have 10 molecules, but actually 8 of them are genes or proteins and 2 of them are compounds. And all of them are very interested in this example, different type of interactions. So something as confirming the example that I was providing to you before, not necessarily if it's upregulated will lead to activation of everything. But some things when a protein is activated or a gene is activated is because something else is less activated, not activated at all, or in specific terms, less abundant or downregulated. So for example, if we take this interaction in here, it says interferes on human GA-PDH mRNA, increased expression of human mRNA. So we can infer that based on when this protein is activated, it leads to the inhibition of this mRNA. The same case with this other interaction, that is the opposite actually, because it's increasing. So when this is increasing, the mRNA of GAPDH increases as well. And that's something of example. But in here, we have, again, multiple examples of the edges that we were discussing in the first video. We have direct relationships, indirect relationships, and PPIs. So this is another way of analyzing data in IPA. As well, another tab that we can search, as we predicted in the first video, we have diseases and functions associated with carbohydrate metabolism, and we can of course confirm what functions are these ones, and we can display this as a network as well, view network. Of course, this is getting more complex, so that's why it's very important to be specific in the networks you want to display, because the bigger the networks, the more convoluted it is to understand and analyze the networks itself. So it's good to work with a small data set, small nodes, so it can make more sense with less. So other feature that we would like to show is, for example, other characteristics and data sets that IPA provides. So if we go to shared projects, shared libraries, and we go to Ingenuity canonical pathways, we have two big pathway folders. So we have the metabolic ones and we have the signaling ones. So let's take an example, apoptosis. So even in apoptosis, we have up to, well, a couple of them, more than 10. And we can, for example, take apoptosis signaling, double click it, and we can see the pathway. And all these nodes. So something very tricky and something very interesting is, well, I have a big data set, or let's say that I have this small portion of data set, and I would like to see if it's associated with some pathway that I want to study because it's of interest of me. Let's say that I would like to study if my proteins are in a way associated with this pathway. So a way to do is to go overlay, analyze dataset list, and the core analysis we run in the first video. Now it became useful as well because now it's not used only for summary, but now it's more specifically in terms of, well, now I just want to work around IPA. Now I just don't conform with what IPA gave me. So now I just want to do something else with the data that I have. So if you want to enrich a pathway with the list of genes you have, you just do this. Select the core. And let's see. Well, actually, it shows that it has something, but it's not significant. Some way that we can check if we have the proteins that are highlighted in gray, that's something important, because in IPA, when you have something that is in red, it means it's because it's upregulated or more abundant. If it's green, it's because it's less abundant or downregulated. But when it's gray, it means that the proteins or genes on your list are not considered significant based on your criteria of significance. So, as well as the nodes are represented in the edges, as well may vary as well. So in the nodes, if it's double, it means that it's a dimer. You have something like a double line between the nodes is because it's a complex, it's a dimer or a complex. So a good way to know which members of that complex are from your data is to double click into the node and highlight show members or membership, and it will display the members that corresponds to that node but also if you want to check which ones specifically are the ones that are in your dataset. In this case, they are in your dataset, but they are not significant. So this is another way to use your data to enrich the pathways that IPA already offers to you. So with this demonstration, I hope all this has been of help to you. And thank you for listening, thank you for watching, and thank you for taking this course.